Thank you, Brett. Good morning, everybody. It's a pleasure to be here. As we're a public company, I draw your attention to the safe harbor statement, as we will be making forward-looking statements, and direct you to our regulatory filings. They're all public, including our recently filed 10-K. We see ourselves to be emerging as a leader in T-cell immunotherapy. We have a lead vaccine candidate entering four phase 2 clinical studies focused on women's cancers, breast and ovarian cancer. We've seen a robust and sustained immune response in phase 1, and we have FDA fast track and orphan drug designation for ovarian cancer. Importantly, the T-cell vaccines that we're dealing with overcome the deficiencies of earlier approaches. Carefully selected antigens derived from patients with cancer generate a memory T-cell response against tumor overexpressed self-antigens. And they address tumors that are not well treated currently and are not adequately addressed by other immunotherapies. And they can be used as a standalone or in combination with other therapies. We see a significant opportunity in women's cancers or women's immune health, simply because these type of cancers are resistant to most immunotherapies. The time to progression is relatively short, and the survival prognosis is extremely poor after recurrence. There's a large U.S. market in ovarian, breast cancer, either triple-negative breast cancer or the larger market of HER2 breast cancer. And these treatments address patients in great need, both to prevent recurrence or to improve outcomes for patients that have undergone frontline therapies. And we have phase 2 collaborations already started with industry and clinical leaders such as the Mayo Clinic, AstraZeneca, Memorial Sloan Kettering here in New York, and the U.S. Department of Defense. The technologies and the targets, we have a multiple epitope peptide vaccines, TPIV200 against folate receptor alpha, which is indicated for ovarian, triple-negative breast cancer, non-small cell lung cancer, TPIV110, 100 against HER2/neu, which is indicated for HER2/neu breast cancer, ovarian, and colorectal cancer. And we have an in-house developed DNA vector platform called Polystart, a very novel platform that can be used for multiple antigens, both self-antigens, combinations of self-antigens, or including neoantigens if we chose to do that. So the approach we have is target-driven. It does not rely on exposing neoantigens. The molecules that are overexpressed occur on the majority of cancer cells. For example, 90% of ovarian cancer cells overexpress the folate receptor alpha. They're discovered through translational medicine. So in other words, they were discovered through observing what cancer patients, the immune system of cancer patients, is looking at. So they've already started to mount an immune response against these antigens. And we're very broadly stimulating both MHC class 1 and class 2 antigens to activate both the helper and killer cells. And we now realize how important it is to stimulate CD4 cells, helper cells. And we are seeing a durable memory anti-tumor response, or we're eliciting it in terms of the longevity of the immune response. And the approach here targets not just the primary tumor, but also metastatic disease, which is ultimately the killer in cancer. It's an off-the-shelf product, easy to produce, low-cost manufacturing. In terms of the phase 1 results for TPIV200, the studies were done at the Mayo Clinic in Rochester, Minnesota, in ovarian and breast cancer patients. The vaccine was safe and well tolerated. Very importantly, over 95% of the evaluable patients generated robust T cell responses. That was a great response to observe. And 100% of those patients demonstrated T cell responses lasting over 6 months, some patients up to a year. The initial findings were published in ASCO 2015. The full publication will be submitted for publication in the very near future. Importantly, these results led to significant collaborations for multiple phase 2 studies. This is a busy slide, but I just want to show the phase 2 studies that are underway. Based on what we saw in the phase 1, the results were good enough, more than good enough to move to phase 2. We have two studies in ovarian cancer, one in platinum resistant. That's a combination study with AstraZeneca's durvalumab, an anti-PD-L1 inhibitor, a checkpoint inhibitor, and that's being done at Memorial Sloan Kettering. The other ovarian cancer program is in platinum sensitive. This is the one that has fast track from the FDA, fast track designation. These studies have already started, and I'll give you a bit more detail on those. And we have one study that started in triple negative breast cancer, which is our own study. It's a dose and boost study. And we have a big study ready to start at the Mayo Clinic in the very near future. That one was fully funded to the tune of $13.3 million by the US Department of Defense, non-dilutive funding. And we'll talk about the HER2 new studies as we move along. This is a busy slide too, but I just want to bring out 1 or 2 points. Anybody that wants to look at the full clinical protocols for these studies can look at clinicaltrials.gov, put in TapImmune, and you can look at all the studies we're doing, all the detail behind it. And so this is a study that I mentioned at the Memorial Sloan Kettering, a combination study of a checkpoint with our TPIV 200. The design of this study is important because the ovarian cancer is not a natural target for a checkpoint inhibitor because it's a low mutation type of cancer. What we are doing here with TPIV 200 is providing the signal for the immune system to see. So it made sense to see if then you could come in and, if you like, take the brakes off the immune system with durvalumab. So it's a very interesting study, even though it's in a very difficult patient population that have failed all chemotherapy. And so this just shows you the study with the different cycles of vaccine and durvalumab, and it also shows you that we're looking at the radiographic assessment of the tumors. It's a 40-patient study. It's open label. Over 50% have already been recruited. It's at Memorial Sloan Kettering plus 6 satellite sites. And we're looking at overall response rate. Importantly, we'll be looking at some interim analysis and a go-no-go decision later this year, around September time, according to the study protocol. The other study that we're doing in ovarian cancer is for platinum-sensitive patients. These are patients that are responding to platinum chemotherapy, will ultimately become resistant. So what we're doing is giving platinum together with the TPIV 200 to see if we can maintain them in a disease-free state for much longer. This is our own study. It's a blinded study. It's 100 patients, up to 15 sites. We've started recruiting for that study. We're looking at time to disease progression and overall survival response rate. The upcoming milestone in this study is the interim analysis which will take place around Q3 2018. This is our triple negative breast cancer study exploring the effects of high dose or low dose. It's a randomized study, and it's looking at also a boost strategy after 6 months. And so the recruitment is for 80 patients. We've already gone past the 25% recruitment mark, where we've received a positive drug safety and monitoring board approval to continue that study. And we expect full recruitment by the end of 2017 at 12 different sites, and we expect at the end of this year to be able to provide an interim analysis on that study. Just briefly mention our plans for the HER2/neu vaccine. We're planning our own study to start this year. We've got a manufactured product, and we're completing our discussions with the FDA. We've had a pre-IND meeting, which was favorable, and so we're just dotting the I's and crossing the T's to get that one started. We recently reported that our colleagues at the Mayo Clinic had got another Department of Defense grant, around $4 million, for a study on ductal carcinoma in situ, DCIS breast cancer, which is the earliest form of breast cancer or precancerous lesions. That study will start very soon, and what we will do in that study, or what our colleagues at the Mayo will do, is apply the vaccine before surgery is done and look at the effect of vaccine on the tumor lesions themselves by imaging. Again, it's nice to be able to get that type of non-dilutive funding in support of our overall clinical program. This is to remind me and everybody that we believe that combination therapies in immunotherapy will be the way that trials will be done, or therapies will be done in the future. And that's because with the success of the checkpoint inhibitors, they're still only reaching a certain level of efficacy. And it will require combinations with vaccines, adjuvants, oncolytic viruses, CAR T cells, to get that survival level at a much higher rate. So there's a lot of effort going on, and including at TapImmune, to look at the best combination strategies possible. And it's important, there are over 800 clinical trials now looking at combination therapies with checkpoint inhibitors, and it's important to have a very solid mechanistic approach to how you do those studies. I'd like to mention our in-house developed technology called PolyStart, because we feel this is a next generation vaccine technology. It's a unique protein expression system that increases the potency of DNA-based immunotherapies. So in other words, it gives a much better presentation of antigen to killer T cells, which should give an enhanced immune response. It's going to drive incremental value for TapImmune. It's proprietary technology invented and known by TapImmune. It has applications for oncology and infectious disease. It has applications, I've already mentioned, to both self-antigens and to neoantigens, and it has high potential for out-licensing, and we're looking for partnerships in that area. And so we're advancing this in our preclinical studies to get it into the clinic to match the current clinical trials we're involved in. We have recent patent approvals that take the patent life of this technology out to 2034. We've recently made new constructs for breast and ovarian cancer. We started out this technology by proof of concept in viral disease, and we're looking for partnerships to continue that. If you look at what PolyStart's about, DNA is attractive as an approach, but one of the problems in this area has been protein expressions being variable to poor. And normally if you have one DNA, it makes one RNA, and you get one protein. In the case of polystart, one DNA plus polystart gives 4 or more proteins, and we've recently quantitated this, and it's at least 4 more. In some cases, it's up to 10 to 12 times the amount of protein. Okay. So this works by using an alternative initiation codon, and it's designed for rapid cut and paste insertion of different antigens. So it can do what we do with peptides, but in a much more interesting way, because you can mix and match antigens, you can get them inside the cell, you can get them put outside the cell. So we believe this is going to be an important technology for TapImmune. We have an experienced management team in terms of product development, finance, and research. Bob Florkowitz here was the inventor of polystart. We have a number of medical and scientific advisors: Keith Knudsen, the inventor of the technology; Edith Perez, who's now at Genentech; Mark Pegram at Stanford, and we'll be announcing shortly a considerable expansion of that group of people. So if I look at our clinical and partnership strategies in 2017, we will complete phase 2 proof of concept studies. We have 4 phase 2s ongoing, around 17 million in undiluted financing, and we'll be having interim results this year. We're obviously seeking pharma biotech partners for late-stage development commercialization, and we'll be further exploring combination regimes just like we've done with AstraZeneca and Memorial Sloan Kettering. And we'll seek to monetize the polystart expression platform. So these are the reasons, I think, why it's important to consider TapImmune as a leading company, an emerging company. We have a lot of things going for us in terms of our pipeline, success so far, vetting by key collaborators. And if you look at our market cap, it's around 40 million. That presents an opportunity. It means there's a lot of room for growth. And so we're excited by what we're doing, and thank you for your attention.